Science Journal for Kids and Teens presents How Much Do Viruses Change Over Time? Read by Miranda Wilson. Abstract. Bacteriophages, or just phages, are tiny viruses that infect bacteria. Some phages help us by killing harmful bacteria and keeping our gut healthy. Although phages have existed for millions of years, we don't know much about the ones from ancient times. To find out, we analyzed super old human poop samples. Using a new technique called de novo assembly, we pieced together the DNA of ancient viruses from these samples. We discovered nearly 300 types of bacteriophages that we had never seen before. But there was also one 1,300-year-old phage that was almost exactly the same as a modern-day species. This was extremely surprising. Our research provides a useful method to rebuild ancient phage DNA sequences and learn more about the history of viruses. Introduction. Ever wonder why you need a new flu shot every year? It's because the flu virus keeps changing itself all the time. In fact, all viruses make small, random changes in their genomes when they copy themselves. These changes are called mutations. Over just a few years, a virus can mutate to be very different from the original. But what happens after 100 years, or even 1,000 years? Bacteriophages are viruses that infect bacteria. These viruses are very important to our health. Your gut has trillions of phages. They can affect both good and bad gut bacteria by killing them or even changing how they work. We wanted to know what the phages in the gut of ancient humans looked like. Identifying ancient phage DNA is difficult, though. DNA itself is very fragile, and it gets more damaged the older it is. This makes old DNA very difficult to work with. Plus, in a sample, the DNA of phages gets mixed up with DNA from bacteria and other tiny organisms. Luckily, other scientists developed new computer programs to fix these problems. These tools can help us understand how ancient phages compare to modern ones. In this photo, you can see many bacteriophages attaching to the cell wall of a bacterium. The black and white image shows the large bacterial cell on the right, and numerous phages can be seen along the outside of it. Methods. In just 1 gram of human poop, there are over 100 billion bacteria and up to 1 trillion phages. This means ancient human poop samples are perfect for finding phage DNA. We picked 30 ancient poop samples. The oldest sample we chose was from a 5,300-year-old frozen mummy named Otzi the Iceman. We also used poop from ancient humans all over the world, including the USA, Mexico, and Austria. And guess what? We didn't have to collect any samples ourselves because they had been studied by different groups previously. We just recycled their data. In a sample of ancient poop, there is a mixture of lots of different DNA. Human DNA, bacterial DNA, and phage DNA are all mixed together. We needed to separate and split this DNA mixture into separate genomes. Usually, we use reference genomes to help with this process. This is like assembling a puzzle using the picture on the box to guide us. We can't discover new species using reference genomes, though. Instead, we use a technique called de novo assembly. This allows us to puzzle the genomes together from scratch with no picture to compare to. Then we sorted out the viral genomes. A few AI programs helped with this. Together, they identified 298 good quality virus genomes from over 35,146 genome fragments. Here in Figure 1, you can see locations where the ancient human poop samples came from. They are marked with red circles and can be found in the USA, Mexico, and 2 different locations from Austria. The large land masses are in purple on the map. On the x-axis, you can see the longitude, and on the y-axis, you can see the latitude. Results. On average, the ancient virus genomes we studied were only about 40% similar to their closest modern-day relatives. That's a big difference. It's possible that many of these viruses have changed a lot over thousands of years, evolving into new forms. Of course, there could be modern viruses that are more similar to these ancient ones, but we just haven't discovered them yet, since there are still many unknown phages. One of our findings really surprised us. Out of the 298 ancient viruses, we found one that was almost identical to a modern virus. This ancient virus was 97.7% identical to a modern virus named Mushu virus Mushu, even though it's 1,300 years old. Even if we consider the slowest mutation rates for viruses, we didn't expect any ancient viruses to be that close to a modern one. Here in Figure 2 shows the similarity between the ancient and the modern Mushu virus genomes. On the top, you can see part of the ancient virus genome, and on the bottom, you can see part of the modern virus genome. Pieces that are similar between the two can be seen in purple, and pieces that are different can be seen in red-orange color. Looking at the figure, what do you notice about the location of the mutations from the ancient virus to the modern one? Do the changes seem to be mostly in one area or spread out equally? Discussion. Our method lets us find phage DNA in ancient samples. Most of the phages we found were different from any viruses we've seen before. Even so, one of the viruses was incredibly similar to the modern Mushu virus, which still lives in the human gut today. We think a reason for the similarity is because the virus survived as a very stable prophage. Some phages have a special ability to insert their DNA into the DNA of their host bacteria. When a phage does this, it becomes a prophage. This is how phages can change the inner workings of bacteria. It also allows them to remain safe for a long time. The prophage does not mutate much because it is not replicating itself directly. This might explain why the ancient virus and the modern one are so similar. Our research made many interesting discoveries about ancient gut phages, but we still don't know much about ancient viruses in general. We hope that our research is the first step towards understanding the long shared history of humans and viruses. Conclusion. Just like us, ancient humans had bacteriophages in their guts. Those phages, along with gut bacteria and other microbes, made up their gut microbiome. The gut microbiome is important because it helps with digestion, supports the immune system, and keeps harmful bacteria in check. A healthy gut microbiome can help us stay well and feel good. You can keep your gut microbiome healthy by eating a balanced diet with fruits, vegetables, and other foods with fiber, drinking plenty of water, getting 8 to 10 hours of sleep, and moving your body regularly. Thank you for listening to this recording. This work has been adapted from the original peer-reviewed paper in the journal Nature Communications, published on January 23, 2024. Research conducted by Piotr Rozwalik, Bas E. Dutil, Andrzej Żelazinski, and others from the Department of Computational Biology at Adam Mickiewicz University in Poland and the Institute of Biodiversity at Friedrich Schiller University Jena in Germany. See the full list of authors and their affiliations in the accompanying PDF. Please visit our website, sciencejournalforkids.org, for more free science teaching resources.